Hello, my name is Jonathan Castillo. This is my Case Connection Unit 2 assignment video and script. I chose to answer Case 1 and Case 2. I will go over Case 1 first. Case 1. Pyruvate kinase is the final step in glycolysis and is primarily regulated by activators and inhibitors under normal conditions. There are two key steps in which pyruvate kinase is normally activated in cells. The first step involves the allosteric activation of the pyruvate kinase enzymes. For M2, L, and R pyruvate kinase and isoenzymes, activation is done by binding to fructose 1,6-bisphosphate. This promotes tetramerization, which forms a tetramer out of the pyruvate kinase isoenzymes. The M2, L, and R pyruvate kinase isoenzymes are then allosterically activated. It is important to note, though, that the pyruvate kinase isoenzyme M1 does not bind to the fructose 1,6-bisphosphate. This is due to having a high constitutive activity and structural difference in comparison to the other isoenzymes. The second step of normal pyruvate kinase regulation can then occur, which involves phosphorylation. The activated pyruvate kinase isoenzyme are used as a catalyst to transfer a phosphate from phosphoenol pyruvate to ADP to form ATP and ultimately pyruvate. But in the process of regulating pyruvate kinase, there are several ways in which the enzyme activity is also inhibited. One way is a direct product of phosphorylation, and as an increase in ATP allosterically inhibits the L and M2 isoenzymes when energy is high. This allows for enough ATP to be formed and for production to stop when enough ATP is formed. A second way in which pyruvate kinase is inhibited is due to alanine and phenylalanine. Phenylalanine binds to the M1 and M2 isoenzymes, thus allosterically inhibiting and decreasing the affinity of pyruvate kinase for phosphoenol pyruvate. Alanine, though, is only able to allosterically inhibit the M2 isoenzyme, but not the M1 isoenzyme. Thus, a negative feedback loop is formed, inhibiting pyruvate kinase. The preferential expression of pyruvate kinase type M2 over pyruvate kinase type M1 involved in cancer cells is mainly due to how the isoenzyme M2 favors anabolic metabolism. The M2 isoenzyme is regulated by the binding of tyrosine phosphorylated peptides. This can allow for growth factor initiated signaling to glycolytic intermediates into other biosynthetic processes. The diversion of these glycolytic intermediates can then be used for such processes like the pentose phosphate pathway. Anabolic metabolism can occur through the pentose phosphate pathway that directly supports cancer cell development. Thus, even though the M2 isoenzyme does not seem to be the ideal isoenzyme to be used, cancer cells promote the expression of the M2 isoenzyme in order to divert substrate into a pathway that supports the cancer cell development. Case 2. Muscle glycogen is mainly used to provide glucose 6-phosphate for glycolysis and ultimately produce ATP for muscle contraction. This is mainly done through the degradation of glycogen through a process called glycogenolysis that involves several steps. First, glucose 1-phosphate will be released from stored glycogen using the enzyme phosphorylase kinase. specifically the active phosphorylase A form. It should be noted, though, that there is another form of phosphorylase kinase, the inactive phosphorylase B form. There are several activators and inactivators that help to regulate the activity of phosphorylase kinase. Usually this means shifting phosphorylase kinase into either the active or inactive form. During exercise, the active phosphorylase A form will be primarily used to provide energy using several activators. One such activator is an increased concentration of AMP. Phosphorylase kinase is cyclic AMP dependent, and thus an increase in cyclic AMP catalyzes phosphorylation via ATP to shift inactive phosphorylase kinase B form to the active phosphorylase A form. Epinephrine is also an activator of phosphorylase, as it signals for an increase in cyclic AMP formation for rapid muscle activity. Calcium also acts as an activator of phosphorylase. This is mainly due to high concentrations of calcium binding to inactive phosphorylase B form and shifting it into the active phosphorylase A form. Once glucose 1-phosphate is freed, another enzyme called phosphoglucomutase is used to isomerize glucose 1-phosphate into glucose 6-phosphate, which will be used for glycolysis. So having a deficiency of muscle glycogen phosphorylase will influence such activities like taking long walks. This deficiency is called McArdle's disease, or glycogen storage disorder type 5. This condition is characterized by having a deficiency or complete absence of glycogen phosphorylase. Common symptoms of this disease do include pain and weakness in muscles, which are usually worse when exercising. But this does not mean that a person can no longer do such activities as walking. It is possible to do moderate intensity aerobic training, which can increase muscle oxidative capacity over time. Studies have shown that this is possibly due to something called a second wind phenomenon, in which taking a brief rest period after about 10 minutes of continuous exercise help reduce exercise-induced muscle pain. So it is possible to take longer walks in time. Also, it is not necessary to get up in the middle of the night and eat food to maintain blood glucose levels. This is because the process of maintaining blood glucose levels is different than that of skeletal muscle glycogenolysis. This is primarily done through the liver, which provides glucose to maintain blood glucose concentration. The process is like what occurs in the muscle, as phosphorylase kinase A form is used in the liver. So a deficiency of phosphorylase kinase would reduce the amount of glucose within the blood glucose concentration, but it is possible that taking in more glucose through diet may help with blood glucose levels. This does not necessarily mean to be done at any specific time of day, but rather the focus is on the type of foods that have more glucose in them. Thus, it is not necessary to eat at a specific time of the day outside of normal meal times. Finally, excessive lactate will not occur during intensive anaerobic exercise with people that have a phosphorylase deficiency. In fact, the amount of lactate produced is less than people without a phosphorylase deficiency, but still comparable to people with the deficiency. Though another reason why there is not an excess in lactate is because people with the phosphorylase deficiency tend to uptake lactate more than lactate is released in the active muscles. This is because their bodies are using the lactate. In the absence of being able to use phosphorylase, the enzyme lactate dehydrogenase is instead used to convert lactate into pyruvate for the TCA cycle. The bodies of people with a deficiency then are compensating by utilizing the energy produced by the lactate instead of anaerobic exercise. Thank you.